Hello, Year 8. So today we're gonna go through using a light microscope and looking at your own cells, animal cells, because we're all animals, under the microscope. So we've got various bits of equipment. Here's my microscope. I haven't actually plugged it in yet, so you need to make sure you're plugged in, because that gives us our light source. Okay, so there we go. Around the back, turn it on, and again have a look at the brightness of the light. You want about halfway, not too bright, not too dim. Okay, then make sure use the coarse focusing wheel. Can we see this? Just move it. Yeah, okay, there we go. The coarse focusing wheel to move the stage all the way down to the bottom. All right. Should be stored, when you store your microscope, store it under 4x magnification. So whenever you start or finish using your light microscope, you'll have it on the lowest magnification setting. All right, so please just make sure of that. Equipment, apparatus we need, here's our mounting needle. That's to help us put the cover slip on the slide when we're making the slide of our own animal cells. There we go. We have a cover slip. That's those two work together. We use the mounting needle to help the cover slip just settle down gently onto the slide when we're making it. There's our slide. So I'll put that on the table there, ready to go. And here's our dye. It's called methylene blue. It will turn the animal cells blue. You remember we used a different dye for plant cells. We used iodine. So this is much better for looking at and staining animal cells under the microscopes. That's why we're using a different one rather than iodine. Okay, so to sample your own cells, you'll need cotton bud. Make sure this has been sterilised and it's inside a nice sterile container. Should be inside a Petri dish that you've been given. Okay. Especially these days, what we're going to do is make sure when you do swab inside your cheek that immediately this is disposed into Dettol for obvious reasons, just to stop any sort of cross-contamination. And you are going to have to take your mask off very briefly to do this. I haven't got my mask on because there's no one else in the room, so let's just try and demonstrate. Maybe I could show you. Okay, mask on. Okay, now I'm ready. Got my swab. Okay, now I've got enough cells on there. I can just pop my mask back on. So on the tip of this now is animal cells, or cells, my own cells from inside my cheek. So I'm just going to sort of smear those cells on to the central part of that slide. Can you see? Okay, so here's the middle, and I'll try and smear about the same size as my cover slip. Yeah, I could do it in a square shape if I like, and smear as much as I can on. Okay, and that's going to go straight into, I've got some disinfectant here, just pop it straight into the disinfectant. Okay, I'll take my mask off just so you can hear me properly. Okay, so now I've got my cells on the slide. What I need to do is one drop of methylene blue. I don't need too much at all. One drop should be enough. Okay, here's my slide. If you want to make sure these are clean, you can just clean them with a bit of paper tissue. Get it from the corner of the science lab and just make sure they're nice and clean. But these are very fragile, so be very careful. So I'm going to set this at an angle so it's like that, and then I'm going to use a mounting needle just to drop it and lower it down onto the methylene blue. Okay, and what that's going to do is stop formation of too many bubbles so we don't view bubbles underneath the microscope. Okay, so gently lower it down. It should then have a slide. Looks a little bit blue, and underneath we'll be able to observe our own animal cells. So put it on the stage, put it under the clips, adjust the position so your light is shining through the blue cover slip area, and then what we're going to do is use the 4 times magnification to search, focus first and then search for those animal cells, and they'll just look like, hopefully your teacher will be able to project something on the board, little blue dots to start with. So you'll be trying to focus on little blue dots that you can see under the microscope, and then we'll increase the magnification to 10 times and see if we can see them a bit clearer and see some structures inside. Okay, hopefully that was useful. Please see my other video on how to use a light microscope to do the focusing. Okay, thank you.